Hi students, in the last class we discussed about microbes in human welfare. Within that case we discussed about microbes in household products, and today class we are going to discuss about the role of microbes within industrial level or within different types of industries. Not only in industries, and we will discuss about different types of the production, right, which are also used in the daily life. Means what I'm trying to say here, whatever that microbes, some of the microbes are used in the industrial level, and some of the microbes produces some of the products. These products also we are using in our daily life. For suppose antibiotics are there. Antibiotics are we are using, right? That is one of the miracle for the humans. During the Second World War, the antibiotics are, especially that penicillin antibiotic was identified by the Alexander Fleming scientist, right? That is the very important product produced by the fungi, right? And see, I like to discuss about some of the products also produced by the different types of microbes, which are also used in the medical field. We'll discuss now, right? See, especially here, industrial level production. One is the fermented products. See, beverages. The main crucial role is here, beverages. See, we know. I thought I already explained so many times what is fermenter. These are the large vessels. Inside, right? There is a sterile conditions are maintained. This is one of the fermenter structure. These are the large vessels, and inside sterile conditions are maintained. Means, there is no chance for the growth of unwanted microorganisms, right? And optimum pH is maintained, optimum temperature is maintained, and all the favorable conditions which are required to the desired microorganism cultivation. Okay, all these conditions are maintained. So these are called as the fermenters. I already said in the biotechnology chapter there is a difference between the fermenter and the bioreactor. Cultivation of eukaryotic cells within large containers are called as bioreactors. Cultivation of the prokaryotic cells within that large containers, these are called as fermenters, right? This is one of the best example fermenter, because inside here we are cultivating that one of the E. coli bacteria or any other bacteria. So this one is called as fermenter. If we cultivate any eukaryotic cells for the hormones production and different types of productions, so that one is called as bioreactor. Maintenance of bioreactor is costly than maintenance of fermenter, because bacteria are very easily cultivated within very cheap cost, but the cultivation of eukaryotic cells is quite difficult. Okay, we need to maintain all the conditions and all the nutrients and wide range of nutrient components are required for the cultivation of the eukaryotic cells. That's the reason why maintenance of bioreactor compared with that maintenance of fermenter is costly, right? Is costlier than the maintenance of fermenter. So simple, bioreactors maintenance is costly, fermenter, okay, which are cheaper than the bioreactors, right? So whatever that the raw material is, one of the fruit pulp is there and malt grains are there, whatever the fruits, see, we know this is a quite interesting chapter, quite interesting topic, but most of the students are well known about the beverage products. I thought I'm expecting. See, there is an alcohol production is there. That is nothing but ethanol production, right? See, one of the within our state also so many number of beverage companies are there. We know, I thought some of the students are well known about the types of the beverage products. There is an brandy, whiskey, rum, gin, right? And wine is there. Sir, why these names are different? All these are produced by the fermentation. Why the names are different? Why the names are different? Remember, all of you are not talking about the company names. I am talking about the basic, which one is called rum, which one is called beer, and which one is called wine, like this I am talking, right? I am not talking about any brands here, right? Remember, see, what we are taking that fruit pulp is there, or in certain cases we are taking that malt beans, okay? See, fruit pulp and malt beans we are taken as the raw materials and which are subjecting for the fermentation process within the fermenter in presence of the yeast, After certain time period, we get the fermented product. Then the fermented product, okay, in two ways are there here with the distillation. If the fermented product is subjecting for the distillation, right, within that, okay, distillation process, okay, or the fermented product is subjected for the distillation. In that case, okay, some components are produced here. Brandy by the distillation, whiskey by the distillation, rum is by the distillation. So why in some cases brandy, why in some cases whiskey, why in some cases rum? See, if the raw materials are changed, for some raw materials, presence of yeast brandy, for some raw materials, presence of yeast whiskey, for some raw materials and yeast is rum. Here raw materials are changed, product is changed, but organism is similar. Right? And fermented products, again here, without distillation process, beer production, right? There is in some of the breezes also there, right, sir? Beer production is there, right? And wine production, the beer and wine production is done by without distillation. After the fermentation process, which are purified but which are not subjected for the distillation process. That's the reason why these are the products which are produced without distillation, beer and wine products. Sir, why yeast have the capacity to produce the alcohol? Why not some other fungi not have the capacity to produce the alcohol? Why yeast have the capacity to produce the alcohol? Okay, why not other fungi? I'm talking about fungi. Why not other fungi? Yeah, produces alcohol. Means only yeast produce the alcohol. Remaining fungi are not producing the alcohol. Majority of the cases. Why? Because whatever that yeast contains genes are there, that genes contains especially that the fermented enzymes are there, right? The fermented enzymes like the zinase enzyme, right? And definitely a group of enzymes which are act on the carbohydrates. And there is an alcohol dehydrogenase enzyme is there, right? These enzymes are accumulated and these enzymes are continuously produced by the yeast cell genetic material. That's the reason why yeast have the capacity. Right? So that is about that fruit pulp, malt grains substitute for the production of brandy, whiskey, rum with distillation. Without distillation is beer and wine. See, we have to know here some of the wines are very costly due to the reason is that there is a process is there. Once the wines are produced, after that production, okay, which are there is a process is there which are stored within that wooden cascades. The wine stored within that wooden cascades, right? After that storage process, again the wine is collected and that wine is poured into the bottle. Based on the storage, some of the wines are stored up to 10 years, some are 15, some are 100 years. So based on the storage, the cost of the wine is varied. Remember, mostly the wines are prepared by the uses of the product, especially fruit products. Mostly not the cereals, the wines especially produced by the fruit products, right? That is about the fermented beverages. Sir, why here these are bad? What's the difference between brandy, whiskey and rum? Basic difference here. See, the percentage of the alcohol is different. In certain ratio, you get alcohol percentage. Okay, Okay, certain ratio in gravity and certain ratio in viscosity and certain ratio in ground, right? That is about the beverage products produced by the different type, especially yeast, right? Within industries, And see, this is one of the industrial production. In the last class also we discussed cereals are the wheat, rice, and there is a bread making process subjected by the yeast. Yeast is the best. Okay, yeast is one of the best organism to produce the wide variety of the products. See, within that alcohol production and within that bread baking process, and different types of products are produced by the yeast. That is about the fermented products. Remember here ethanol. Ethanol is nothing but ethyl alcohol. So here ethanol also produced by the fruit pulp and the malt grains. Presence of the yeast. Ethanol is produced. Right? So ethanol is also produced by the distillation process. Right? That is about that fermented beverages. Next one we will discuss about that antibiotics role and antibiotics production. Coming to the next topic is the antibiotic productions. We know the role of antibiotics within the human life. See, definitely antibiotics are very important. Now this situation, we are facing the pandemic situation. The antibiotics are very, very important, but still we don't have the proper antibiotic for corona, right? See, human life, majority of the cases depends upon the antibiotics, no doubt. See, one side is the vaccine, other side is the antibiotics. First of all, antibiotics are very important. Still for some of the diseases, there is no vaccine. Only antibiotics are available. Maybe the virus means viral antibiotics, maybe the bacterial antibiotics, right? See here, the concept is here antibiotics. Antibiotics, the word derived from the Greek. Right? This is one of the Greek word. We know already anti means against, right? Bio means life. So a group of chemical molecules are some of the chemical molecules which are against to the life. Remember, against to the disease causing agents. That is nothing but pathogens. See, the concept is here, whatever that chemical compounds are there, some of the chemical components are against to the microorganisms, means kills the microorganisms. Right? So that is the basic standard definition for the antibiotics. See, the antibiotics are not against to the human beings. For human beings, antibiotics are very, very useful. But whatever that microbes or pathogens are there, or disease causing agents are there, for that agents, the antibiotics kills. Antibiotics kills that kind of disease causing agents. So how the antibiotic have a capacity to kill different types of organisms? How the antibiotics have the capacity to kill different types of organisms? All of you remember, some of the antibiotics are act on the cell wall, some of the antibiotics are act on the DNA, and some of the antibiotics are act on the RNA, and some are act on the ribosomes. So suppose we know the well-known best example is the penicillin. Penicillin is the antibiotic which is especially prevents the cross walls formation. Whatever that NAM and NAC and pentaamino acids linkages are there within the attached cell wall. See, this is one layer and this is another layer. During the cell wall formation, many number of layers like structures are formed. That cross walls formation or the cross linking formation prevented by the penicillin. See, the penicillin, this is one of the fungi. Right? Penicillin is produced by Penicillium notatum. Right? Understand? Penicillium chrysogenum. Penicillium chrysogenum. Right? So that is about that some of that fungal species produces that penicillin antibiotic. Right? And sir, how the penicillin discovered? How the penicillin discovered? See, this is one of the accidental identification. Accidental identification. This one is also. Alexander Fleming is a scientist. Right? He isolated, or he first identifies the penicillin antibiotic during the Second World War time, and most of the American soldiers also protected. That time, that time this is like an miracle medicine. Penicillin is a miracle medicine because most of the American soldiers are affected. Okay, many number of wounds are there. But when the soldiers uses this powder or this antibiotic substance, okay, the wounds are recovered within the short span. The wounds are healed within the short span. Okay, that's the reason why here Alexander got the Nobel Prize. Alexander got the Nobel Prize. See how the discovery, how the identification is done here. How the penicillin identification is done. See, this is the one of that. Just imagine he is working. Alexander Fleming working in a laboratory. Especially he working on the Staphylococcus. Staphylococcus. That is nothing but Staphylococci, not coccus. Staphylococci. Staphylococci. That is one of the bacteria. Okay. Upon that bacteria, Alexander working. Right. See, he cultured, means he prepared the nutrient media and he inoculated that Staphylococci bacteria. Right. After that, after 1 week, okay, he kept the petri plates in one place. Okay, one of the unwashed petri plate, okay, he observed within that unwashed petri plate, at the center, no growth of bacteria. Actually, he is working on bacteria, but that time one of the petri plate at the center is not having the bacterial growth. Then he is very interested with that one because at the center of the plate, why the bacteria are not grown, and he did the research and he concluded within that region one of the mold is grown, right? That mold inhibits the growth of bacteria. That is nothing but that fungi inhibits the growth of the bacteria. Then he did within that region, and again the mold is separated. Again Staphylococcus bacteria cultivated, that mold also added to that media. Again the bacteria are not grown within that region, and he did the research within this area, and finally he isolated what compounds are present, and he given one of the statement, that is the penicillin. Penicillin is the antibiotic, and after that Alexander and some of the scientists, Fleury and Chain, okay, these two scientists continued, okay, on that penicillin work. Then three of them, it's Alexander and Fleury and Chain, okay, both the three members got the Nobel Prize in the 1945. Right? And there is a plague, whooping cough, and diphtheria, leprosy. And these diseases also well controlled by the antibiotics. See, just I discussed here the antibiotics which act on the cell wall, and I discussed here only related to the penicillin. This is the part only related to the NCRT. And this is quite different from this. This is out of NCRT, but this one is also very important. See, some of the antibiotics act on the DNA, blocks the DNA replication process. Blocks DNA What are the enzymes which are involved in the DNA replication process? The enzymes are inhibited. We know that there is a DNA polymerase 1, polymerase 2, polymerase 3, which are especially present in prokaryotic cells. That polymerases are blocked, or in certain cases, topoisomerase enzymes are blocked. Right? Once these enzymes are blocked, there is no DNA replication process. There is no DNA replication process. There is no growth of microorganisms. There is no growth of microorganisms. There is no disease. There is no disease. Right? Coming to the next one is the RNA. See, RNA means transcription inhibition. Transcription inhibition. The transcription is inhibited. The transcription is inhibited here. How the transcription is inhibited? Simple. See, RNA polymerase enzyme is blocked. RNA polymerase enzyme is blocked. Once the RNA polymerase enzyme is blocked, there is no the transcription process. There is no mRNA production. There is no DNA production. Right? And the ribosomes are blocked. Maybe the 30S subunit is blocked, or maybe the 50S subunit is blocked. See, there is a tetracycline there, and some of that ampicillin, we know about that ampicillin, right, which are especially act on the ribosomes. Rifampicin is act on the RNA synthesis process. Means rifampicin inhibits the transcription process. Right? Just these are the examples. Don't worry about to write about these antibiotics. Just I'm saying this is out of NCERT concept, but very important to remember. So, sir, if the DNA is blocked, if the RNA synthesis is blocked, if the ribosomes are blocked, why the microbes are killed? How the microbes are killed means by the blockage of DNA, RNA, and the ribosomes, the microbes are killed. Sir, if these are blocked, how the microorganisms will be died? How the microorganisms will be died? Simple. We are blocking the gene expression process. We are blocking the gene expression process. For the bacteria cell division, requires DNA replication. We block the DNA replication. We block the DNA replication. There is no new bacteria production. We block that mRNA production. There is no proteins production. There is no function. We block the ribosomes. There is no protein production. There is no function. So these are also very effective antibiotic components compared with the penicillin. Nowadays, majority of the people are targeting the DNA, RNA, and ribosomes of the maybe the bacteria, maybe the some of the fungi or other disease-causing agents. Right? That is about that the antibiotics production.